So this is really a treat. I mean, UCSD is really such a wonderful department of psychiatry, and for me, particularly special. Especially, I have so many dear friends here who have been good friends of mine for decades now. So it's really very special. It's also special because this room, I didn't know I would be speaking in the Marilyn Farquhar room, but Marilyn and George were my teachers in medical school, and George Pilati, her husband, who passed away, and so this is a treat as well. I thought I saw her in the cafeteria yesterday when we were having lunch. And the other thing, of course, is for UCSD, I'm also known as the other Krystal, because my brother has been down visiting a number of times. So I'll be talking today about ketamine, and I'll try to cover two areas of where we've applied ketamine to our human translational research. One having to do with its role or utility as a probe for the neurobiology of schizophrenia, where we've tried to use insights gained from studying ketamine to develop new treatments. And then the other area, which is a very active area of research here, which is the use of ketamine itself as a treatment for depression, as a rapid-acting antidepressant. The deep idea that I'm going to try to suggest today, and hopefully come back to several times in my talk, is that when you study the modulatory systems of the brain, norepinephrine, dopamine, serotonin, you find that there are a number of psychiatric disorders that benefit from that same kind of modulation, say serotonin 2 receptor antagonism, and no matter what the diagnosis is. But as you start to get closer and closer to the biology of that psychiatric disorder, the underlying neurobiology in the cortex, then it starts to make a bigger difference what disorder you're treating. In other words, some categorical as opposed to dimensional features of the biology of psychiatric disorders becomes revealed when you start to target the molecular signaling associated with those disorders. And so, rather than ketamine being universally a treatment for all conditions, I'm going to try to make the case in a way that we've learned something about a striking difference in the biology between mood disorders and schizophrenia through studying the effects of ketamine. These are my disclosures. There's really only one disclosure that's really relevant, critically important for you to understand, which is that our patent for the intranasal use of ketamine to treat depression was licensed by Johnson & Johnson. And so you should keep that in mind as I talk about the antidepressant effects of ketamine. So where are we in the pharmacotherapy of psychiatric disorders? In many ways, we have not had a fundamentally new mechanism for the treatment of either disorder in about 40 years. We know that these medications leave many patients with symptoms that are not adequately treated by anything that we can do currently. And that these treatments are predominantly symptomatic rather than targeting the etiology of the disorder. And as I've mentioned, not really specific to the psychiatric disorders. So, as I've mentioned, I'm going to be talking today about how we've used ketamine to study both schizophrenia and depression. And the conceptual approach that led us to study ketamine in schizophrenia is a relatively simple one. As you all know, the predominant theory for schizophrenia for a very long time was that there were disturbances, in fact, hyperactivity of the dopamine nerve cells projecting to the basal ganglia, and that dopamine hyperactivity could cause psychosis, and you could treat that with dopamine D2 receptor antagonists. When I was starting my career, I had the opportunity to participate in the late, after marketing studies of clozapine, and I had the impression that while clozapine represented an incremental advance in the treatment of schizophrenia, that we needed to probe new biology. I thought that clozapine took us about as far with monoamine signaling as we could go, and I wanted to kind of follow a new path. And around that time, there had been emerging postmortem studies the beginnings of CAT scan and then MRI studies to suggest that schizophrenia might fundamentally be a disorder of cortical development. And if I wanted to study the intrinsic signaling of the cortex rather than studying the modulatory signaling, then I probably needed to either study glutamate signaling or GABA signaling. And the probe that I and my colleagues explored was a probe of the NMDA subtype of glutamate receptor. In other words, something that would allow us to study that aspect of glutamate signaling in people. And this is an illustrative example of a number of studies that we had done with ketamine in healthy subjects that made the point, I think an important point. Here is placebo. In green is a dose of amphetamine intravenously administered. Red is ketamine administered to the same people on a different day. And black, the combination of amphetamine and ketamine. And you see that they all produce about the same severity of psychosis. And there's really not really an additive effect. In other words, they kind of converge, you might say, on producing a certain level of severity of psychosis in healthy subjects. But if you look at the negative symptoms of schizophrenia, the amotivational, asociality, withdrawal, that sort of thing, then you see that amphetamine does not produce these symptoms at all, that ketamine produces these symptoms quite robustly, and that if anything, amphetamine attenuates the effects of ketamine in this dimension. The point is that we have a variety of pharmacologic approaches and to thinking about schizophrenia, but ketamine was providing us the first pharmacologic approach in humans that could encompass a wide range of deficits that we associate with schizophrenia. That if one was interested in a syndrome that had essentially just positive symptoms, that then amphetamine, at least acute amphetamine by itself, could achieve that. In fact, achieve that in a way that's more selective than ketamine. But if one is interested in modeling this entire syndrome, that ketamine might be a useful strategy to pursue this very complex biology of schizophrenia, particularly symptoms like negative symptoms and cognitive impairments, for which our understanding is not very advanced. Now, we started that work around 1989 or 90, and now there's a lot known about the glutamate synaptic biology of schizophrenia, emerging from genetic studies that have implicated consistently glutamate signaling and glutamate synaptic genes and glutamate synaptic development genes in the genetics of schizophrenia. Postmortem studies from groups like David Lewis showing glutamate synaptic spine loss, and NMDA glutamate receptors are right on the tips of those dendritic spines. Had a good fortune to collaborate in a study, in vivo study using a SPECT ligand showing deficits in glutamate receptors in vivo in people with schizophrenia. This was a study led by Lynn Pulaski. In our lab, with a study led by Adil Shavus and with work by Danny Mathalon, could show that an NMDA receptor-dependent form of neuroplasticity, a human analog of long-term potentiation, was deficient in schizophrenia as well. So all of this and more points to disturbances in glutamate synaptic signaling, particularly NMDA-related glutamate synaptic signaling associated with schizophrenia. So that raises the question, well, how do we understand the functional implications of those deficits in glutamate synaptic signaling? And there, we tried to build on the work first of Patricia Goldman-Rikic and now continued by Amy Arnsten, which builds on a very fundamental idea, which is that when you keep an idea in mind, a function, cognitive operation known as working memory, that there are circuits that maintain that information online in an active, excitatory way, what Donald Hebb called reverberatory excitation. And that capacity to retain information online in this excitatory way is dependent on the NMDA receptors on these individual dendritic spines being stimulated. They have the right kinetics as has elegantly been worked out by Xiaojing Wang and other computational neuroscientists, that NMDA receptors have the right kinetics to allow this reverberatory, sustained activity to take place. And so you can show some pretty clear ideas that can translate from primates to healthy humans to patients with schizophrenia. Here is work from Amy Arnsten's lab showing that in layer 3 of the lateral prefrontal cortex in non-human primates, that neurons are activated as information is encoded into working memory, and that activation is maintained to some level while information is maintained on line, and then it drops off. If you iontophoresis, in other words, spritz, a little MK1, an NMDA glutamate receptor antagonist, you greatly attenuate this activation associated with the encoding and early maintenance of information in working memory. Now, we're not going to stick an electrode into a human brain and give people ketamine into their brain in order to replicate that finding. But what we can do with functional magnetic resonance imaging is map the activation of the lateral prefrontal cortex as people are encoding information and maintaining information online. And when we give them systemic rather than a local injection of ketamine, we attenuate the activation in schizophrenia. This observation constrains the interpretation of this observation, and this observation helps to inform the interpretation of these data, which is that healthy subjects, as I showed you before, activate lateral prefrontal cortex associated with the encoding and maintenance of information in working memory. But people with schizophrenia show a deficit in the activation of this region during the encoding and maintenance of working memory that looks a little bit like that. So these kind of data suggest that the similarity between the behavioral syndrome produced by ketamine in healthy subjects is not only a behavioral similarity, but there are also maybe important parallels in terms of cortical synaptic dysfunction. Now, it's important to keep a developmental context in mind, because any fundamental disturbance, any fundamental glutamatergic developmental disturbance is going to produce a compensatory developmental abnormality in GABA systems. So it's not surprising that, again, drawing on a figure from David Lewis, that several GABA neuronal subpopulations appear to develop abnormally in schizophrenia. The somatostatin neurons that are on the distal dendrites engate excitability of pyramidal neurons, CCK neurons, basket cells that gate timing of pyramidal neurons, and parvalbumin cells, which are also basket cells and chandelier cells, which have other timing-related properties as well. So how does the ketamine model help us to understand or probe some of these aspects? And I mentioned the developmental point of view, the idea that the GABA deficits might be secondary to the glutamate synaptic deficits, because so far there aren't prominent implication of GABA-related genes in the heritable risk of schizophrenia. There are some related to neuronal migration, like renin, but not so much. And that we know that NMDA receptor blockade, particularly in development, can have a profound effect on GABA neuronal development. Postmortem studies, though, suggest that these GABA deficits, even if they're compensatory, are present as early in the course of schizophrenia as has been studied. In other words, some studies of a relatively early course schizophrenia. So ketamine recapitulates potentially some of these deficits because it has the ability in certain GABA neuronal populations to block or attenuate the activation of GABA interneurons. When there's a reduction in GABA, there is then a resulting disinhibition within cortical networks, and that disinhibition in the context of NMDA receptor antagonism distorts cortical output. So evidence supporting this model come from a study conducted by James Stone. I had the good fortune to collaborate with him. James is at the Institute of Psychiatry in London. And this was a magnetic resonance spectroscopy study of cortical glutamate levels, in frontal cortical glutamate levels. And here in healthy subjects, you can see the pre-ketamine level, and then they gave ketamine and raised glutamate levels, and the extent to which ketamine produced psychosis in these healthy subjects correlated with the extent to which glutamate increased. So something about this increase in cortical glutamate seems to be a contributory factor to the kinds of symptoms associated with ketamine. And I could give you a whole talk just on this subject, but I'm just going to draw your attention to some other properties, because disinhibition, as I alluded to when I was describing the cortical GABA interneurons, deficits in GABA have more functional implications than just disinhibition in networks. For example, one consequence may be an increase in resting high-frequency oscillations. NMDA receptor antagonists reliably produce these effects in animals. In humans, there are some data from the Santa Cora paper with a drug called linicamine, AstraZeneca drug, AZD6765. We've done some work with ketamine looking at cortical oscillations and with memantine. And there is an interesting and complicated story which is studied intensively here at UCSD by Greg Light and David Braff and others, where there may be an increase potentially in resting gamma oscillations, but some impairment in recruiting in task-related gamma oscillations. And we're interested in whether the possible increases in resting gamma, if they occur, might be analogous to the increase in resting gamma oscillations. We're interested in that because it may help us to understand the nature of what GABA neuronal populations are sensitive to ketamine and which are most important for producing certain kinds of distortions in cortical network function in schizophrenia. So what's known in humans is that the density of NMDA receptors seems to be higher relative to AMPA receptors on the somatostatin subtype of GABA neuron relative to the parvalbumin in basket cells. So that we had expected that maybe the somatostatin cells might be more sensitive. And the logic goes something like this, that if you block the NMDA receptors on the somatostatin cells and you reduce the inhibition of the distal dendrites, then you disinhibit the pyramidal neuron. Okay, so far so good, because I showed you that ketamine can cause a rise in glutamate levels, and I could have shown you a variety of other kinds of data showing that cortical excitability increases when you give ketamine. So the question you might have is, how does ketamine produce the increase in gamma oscillations, the increase in resting gamma oscillations in healthy people, and how does that relate to increases in resting gamma oscillations in schizophrenia? Well, the idea is that if you reduce the somatostatin cell excitability, you in essence disinhibit the parvalbumin neurons, increasing their activity and increasing the expression of at least these high-frequency cortical oscillations that seem to be dependent on them. So that might help to shed some light on this interesting pattern that you see with ketamine and potentially see with schizophrenia. But we've been really interested in another property of these somatostatin GABA cells, and that is that the somatostatin cells not only gate the overall excitability of the neuron, but they actually decide which input gets its way. In other words, that when the somatostatin cells are operating optimally, they enable the glutamate neurons to be selectively activated by specific inputs. And when you disable the somatostatin cells, then what happens is that every input gets its way. And the output, as a result, is really abnormal because no coherent message can get through, and instead what you have is a very dysfunctional hyperconnectivity of this neuron with all of its inputs. And by hyperconnectivity, I mean that this neuron starts to show correlated activity with parts of the brain or neurons that it shouldn't be correlated with under optimal circumstances. So we can look at this a little bit with functional magnetic resonance imaging by creating a profile of the pattern, the level of connectivity of every little box, in this case of the frontal cortex, with every little box of the frontal cortex. And you see that there's a normal kind of distribution. And when we give ketamine, there's a sign that we're increasing the connectivity, the correlation of every little box, because we've shifted the profile to the right. Now you might say, okay, so I've shown some data that suggests that ketamine makes the brain hyperconnected. Well, haven't I by definition now shown that ketamine is irrelevant to schizophrenia, because we all know that there are disturbances in connectivity in schizophrenia? Well, we've applied these same analyses in schizophrenia, and we find that there's evidence of hyperconnectivity, even some signs of it in chronic schizophrenia. But the profile of connectivity is very interesting. So if you look at a high-risk population, and this is work that's been, this is a study by Naomi Driesen in the group. Alan Antichevich has published this work in Biological Psychiatry. So here is a genetically high-risk group in yellow, a first episode group in red, and a chronic group in green. And the chronic group is both increased and decreased, but there's a hint that with the progression of illness that you see some shift from hyper to hypoconnectivity in terms of overall covariance of voxels in the prefrontal cortex. This raises an issue of whether or not this pattern of functional connectivity is a functional sign that fits in with other evidence of progressive pathology in schizophrenia. So, for example, Marsman and colleagues had a really nice meta-analysis of spectroscopy studies that they published in 2011 that showed that cortical glutamate levels were elevated early in the course of schizophrenia, but deficient in chronic schizophrenia. Scott Small's group, in an elegant paper in Neuron in 2013, showed a transition from hypermetabolism in people who had not yet converted to schizophrenia towards structural and metabolic deficits in the hippocampus when they manifested psychosis. And I think we're probably all well aware of MRI studies and DTI studies suggesting that aspects of the gray matter and white matter volume deficits may progress in schizophrenia, particularly the elegant studies that Jay Gee did and colleagues did at NIMH on the childhood onset schizophrenia. What's interesting is that NMDA receptors have the same kind of pattern effects. NMDA receptor antagonists produce the same kind of pattern effects in animals and humans. In other words, that they initially produce hyperactivity, functional connectivity, but chronic, high-dose, steady administration in both animals and humans reduces functional connectivity. And in humans, high-dose ketamine abuse actually produces some symptoms associated that have a factor structure that is very similar to what you see with schizophrenia. So in summary, I've described some effects of ketamine in healthy subjects that resemble network disturbances associated with schizophrenia. And I've made the case that the disinhibitory effects of ketamine, producing hyperconnectivity, may be particularly important early in the course of schizophrenia. And that there may be a response to that, or certainly a shift, but maybe as a response where the network detects the hyperconnectivity and the shift in EI balance, which may very well trigger a loss of functional and structural connectivity. This work this hypothesis arises from the work of a number of people who have written about synaptic homeostasis, people like Gina Turgiano at Brandeis, Eve Marder at Brandeis, and Gray Davis at UCSF, showing that networks really do monitor excitability very carefully and will downregulate over time. And it's interesting that chronic ketamine effects have a number of neurobiological correlates that resemble chronic schizophrenia more than early course schizophrenia. So if I put this another way, I wonder whether it would be a little clearer. So the idea is, again, that schizophrenia starts out at birth as a disorder of glutamate synaptic dysfunction. So that there is a glutamate signaling deficit which is registered in these local networks, and that there's an initial allostatic, I use the phrase allostatic meaning that there is a downregulation of GABA which restores functional balance between excitation and inhibition. But there are consequences related to the ability to tune cortical network function, disturbances in cortical oscillation, and hyperconnectivity. And also, so you have two kinds of adaptations. One is that you have this downregulation of GABA to raise excitability. But this is also occurring at a time when synapses are proliferating widely through the brain. And it's not until the teenage years that you hit a programmed synaptic elimination. And that programmed synaptic elimination starts to correlate with the emergence of the syndrome, synaptic downscaling, and you have the second level allostatic adaptation, where you go from deficit in signaling to hyperconnectivity to synaptic downscaling. And this synaptic downscaling augments the natural pattern of synaptic elimination that takes place in late adolescence, so that you have a double whammy, if you will, an adaptive synaptic downscaling augmenting the impact of synaptic elimination. The point that I want to make by this kind of elaborate scheme that I'm presenting here is that the development of a single medication that would target the underlying biology of cortical deficit in schizophrenia and cover all of these phases of illness starts to be really hard. And this may shed some light why it's been so difficult to come up with a single medication that had a large impact on the biology of schizophrenia. And what I'm going to suggest is that maybe we need to think about targeting particular phases of the illness with particular kinds of treatments. So that's what I'm going to finish up the schizophrenia section of my presentation by talking about. So the simplest idea would be that if you had deficits in NMDA glutamate receptor signaling, then why don't you just stimulate the receptors and somehow ameliorate right at the start glutamate synaptic deficits. Now, one strategy that we and others are looking at are the glycine transporter inhibitors. It turns out that GlyT1 doesn't have a favorable distribution. In other words, it's particularly dense in the brainstem and lower areas of the brain. And it may not be the optimal target, although we're holding out some hope for this. Initially, there were positive studies from Roche that were consistent with some encouraging data with drugs like glycine and D-serine. But one interesting thing that we found, this is a study by Scott Woods, that if you use these glycine-like substances in the prodrome, then they may be helpful as monotherapy, whereas in chronic schizophrenia, they're only useful as adjunctive therapy to antipsychotics. So it may be that if you get people where their synaptic signaling is relatively intact and you enhance NMDA receptor signaling, that you can have a bigger impact on restoring functional connectivity. But what we're really kind of hoping, and this is a strategy that I know that you all are interested here, is the idea that you could enhance neuroplasticity with GlyT1 and then remodel the synapses through experience-dependent plasticity using things like cognitive remediation. And we're currently doing a study like that. The other hypothesis that comes out of this ketamine work is that that disinhibition may be particularly important to treat relatively early in the course of schizophrenia. And that later on, that the same medications that suppress hyperconnectivity and disinhibition might exacerbate deficits in synaptic connectivity and might not be good. So that inhibitory treatments might play a role in schizophrenia, but only during a particular phase of illness. And the treatment we've been most interested in are mGluR2, metabotropic glutamate receptor 2 agonists or positive allosteric modulators, building on the work of Bita Moghadam and Daryl Shep. And there was a very interesting positive study, which was the first non-D2 blocking drug to look like it worked in the treatment of schizophrenia, and it is perfect in every way except one, which of course is they couldn't replicate this finding once they found it. Yeah, which let's let's not get picky about the but they went back and they did a very interesting secondary analysis of their data, which supports in a way the model that I was suggesting. So here in blue is Risperidone. It worked as expected. Here in red is the 40 milligrams dose of the mGluR2/3 agonist. In green, you see if you go to a high dose that you don't get any activity. Perhaps that's excessive inhibition. But this is very promising for the low dose in the early course schizophrenia patients. You would predict in the chronic patients that you would lose the efficacy of the low dose, which they do. But equally important from the perspective of the heuristic model is that the ineffective dose, the high dose, now makes patients worse. So this is a hint that maybe in chronic patients, a drug that reduces cortical excitability to this extent worsens network function rather than helps it. So, in summary, what I'm suggesting is that glutamate synaptic deficits, which are probably present from birth, before birth, probably present as soon as those aberrant genes are expressed, could be a target potentially for prevention. And the question is, do we need a drug to engage these targets for prevention, or could we use environmental stimulation for at-risk populations to try to compensate if we knew who that group who had maybe a 10 or 20% increased risk of developing schizophrenia? Maybe we do need more focused intervention. As people move into the prodrome and symptomatic prodrome, that disinhibition is probably a very important secondary deficit that needs to be treated. And that we may be able to use drugs like lamotrigine or mGluR2 agonists or other maybe low doses of AMPA antagonists to attenuate that hyperconnectivity. And then we have this elephant in the room. And I think we have to be honest that we have no treatment. We have no treatment for the synaptic connectivity loss in chronic schizophrenia. And if we acknowledge that, maybe that'll help us as a field to try kind of neurotrophic therapies or other out-of-the-box treatments that might help to regrow synapses and, in fact, dendritic fields that have been lost through this developmental process. So this is kind of a good news, bad news. Hopefully this is the good news. And hopefully this is good news in the long run, too, as we begin trying to address the full profile of schizophrenia. I'm going to move on to now talk about the ketamine work in depression. And there's some of you may know the story, Alice's Restaurant, Arlo Guthrie. And Arlo Guthrie in Alice's Restaurant goes through a very long story. And at the end of that story, he says, I told you that story so I could tell you this one. And so I told you that story in schizophrenia because in schizophrenia you've gotten to understand that there's a problem of deficits in NMDA glutamate receptor signaling and disinhibition in cortical networks that constitute targets for treatment. I'm going to tell you now that schizophrenia, depression, may be the opposite of that. And the path that took us there was that the field of depression was also focused on monoaminergic systems, in this case projecting from the midbrain for serotonin neurons and pons for norepinephrine, and that the field had focused almost entirely on the monoamine signaling mechanisms and almost completely ignored the intrinsic signaling of the targets of these monoamine systems. In other words, again, glutamate and GABA. And one of the reasons that we were interested in making this shift was that Dennis Charney had led a heroic series of studies at Yale, first depleting serotonin in healthy subjects and they didn't get depressed, then depleting norepinephrine and dopamine with AMPT in healthy subjects didn't get depressed, and then, most remarkably, depleting norepinephrine, serotonin, and dopamine by combining AMPT and tryptophan depletion, and they still didn't get depressed. They weren't happy, but they were not depressed. So that made us think that maybe the intrinsic signaling was really very important. And there was this very intriguing literature that goes back to the history of psychopharmacology, the archives. And you may remember that the monoamine oxidase inhibitors were discovered in tuberculosis patients because a drug called iproniazid was used to treat tuberculosis, and then Nathan Klein found that iproniazid worked to treat depression in these tuberculosis patients. There's actually a parallel story from one of the lifelong enemies of Nathan Klein named George Crane, who studied a different anti-tuberculosis medicine called seromycin and found that patients with tuberculosis who got seromycin had an alleviation of their depression. And then he did a study where he gave seromycin, also known as D-cycloserine, to depressed patients, and they got better. And in this 1961 paper, he says, it's difficult to explain why psychiatric benefits should have occurred almost immediately following drug administration. Really interesting. Ignored for about 30 years after publication. But the thread was picked up for completely different reasons by Phil Skolnick and his colleagues, who were studying a glycine partial agonist called ACPC. And the idea was that this drug might enhance neuroplasticity and might thereby enhance the ability of animals to be resilient to the effects of stress. And when he found that ACPC worked to alleviate depression in this animal model, he went on to study NMDA receptor antagonists and showed that they also produced rapid-acting antidepressant effects in the animals. So we decided we would use ketamine to probe glutamate signaling in depression. And we generated these data, which we presented in 1997 and published in 2000. And with this very interesting observation that while ketamine acutely produced modest symptoms of psychosis that were transient, euphoria and transient, that after these symptoms went away, and we were getting ready to do our safety discharge, our debriefing as we always did at the end of every ketamine day, that some of the patients started to report that they were feeling better. And that by the next day, there were several people who, essentially half the sample that had responded, and many of them really felt like they were, if not completely better, better in a deeply meaningful way. This was a very surprising finding to us, shocking in some ways. And so it's been really wonderful to see that across the studies that have studied ketamine, that the profile of response has really held up. And so that I think at this point there's little doubt that a single dose of depression produces clinical response in somewhere between 50 and 75% of typically treatment-resistant depression. One of the questions about the ketamine response is whether it is because there's a strong placebo response. And so some studies have tried to compare ketamine to medications that we know have some active effects on mood, like midazolam. And what you see is that ketamine produces a more profound improvement in mood relative to midazolam, and this effect persists much longer in ketamine than in midazolam as well. Now you have experts in ketamine infusion for depression here, David Feifel and Scott Irwin, and they will tell you that ketamine is very safe. It seems to be effective in depression, even depression that has failed to respond to electroconvulsive therapy. I've even heard of a case from Helen Mayberg of someone who failed to respond to deep brain stimulation who responded to ketamine. It seems to be remarkably effective for suicidal ideation. I'm not sure if this is because of a distinctive effect on suicidality, or rather that suicidality is a marker of relatively more severe depression. It's effective orally, intranasally, intramuscularly, subcutaneously, and one of the products that I mentioned before, as ketamine, is now in phase 3 trials as an intranasal preparation. It's interesting that ketamine is antidepressant in patients, but it is not mood elevating in healthy subjects. In other words, if you're discouraged but not depressed, you're having a bad day, and you take ketamine, the next day you are not going to feel at the top of your game better than you ever felt before. What you're going to feel most likely is hungover. And that's because ketamine, it has some effects in terms of hangover that are similar to another NMDA receptor antagonist called ethanol, which also produces a hangover. And another interesting thing about the biology is that we've had a series of cases of psychotic depression that respond to ketamine. But if you give ketamine to other forms of psychosis, like schizophrenia, it worsens schizophrenia. So it's not about the psychosis, it's about the depression. And it's not about elevating mood, it's about the synaptic pathology underlying the illness. And we think that it works, and this is obviously an oversimplification, and there's a lot of new work in this area, in part because it stimulates glutamate release, causing enhanced signaling via the AMPA receptor, raising BDNF levels, stimulating downstream signaling via TrkB receptors, and enhancing signaling via mTOR, which drives dendritic spine growth. And we think that because if you block AMPA receptors in animals, you block the antidepressant effects of ketamine. If you block mTOR with rapamycin in animals, you block the antidepressant effects of ketamine. But there's another story this is work that emerged from Ron Duman's laboratory. There's another story that first emerged from the work of Hardingham and colleagues and later developed by Lisa Monteggia and her colleagues at University of Texas Southwestern Medical Center, which is that there are extrasynaptic feedback mechanisms via extrasynaptic NMDA glutamate receptors bearing the NR2B or GLUN2B subunit, which when stimulated activate elongation factor 2 and suppress BDNF levels. So ketamine in essence does two things that are antidepressant. It stimulates glutamate release via this GO pathway, and it blocks overstimulation of this stop or shutdown pathway. And because it does both of these at the same time, we think it's particularly potent and rapid acting as an antidepressant. Ketamine is now being used to explored in other disorders. We have not found it to be particularly useful in OCD. Carolyn Rodriguez at Columbia has more encouraging data there. Dennis Charney has found evidence that ketamine may attenuate symptoms more effectively than midazolam for PTSD. In this PTSD data, they removed the depression symptoms, or covariate, for the depression symptoms. So it has this effect above and beyond the antidepressant effects. It looks like ketamine effects, as you probably know, given David's work here and Scott's, that repeated doses of ketamine appear to be able to sustain the antidepressant effects. And that there are a variety of other drugs that are now being studied that may have the possibility of reducing the abuse liability or dissociative potential associated with ketamine. The drug that's farthest along in phase 3 is S-ketamine. So, what do you do with a rapid-acting antidepressant? And one of the ideas which could have a profound impact on our society and what it means to be depressed is that you have a high probability, if someone is in a severe, disabling, life-threatening depression, of getting them better. When I say high probability, I mean 50 to 75%. Think about that in comparison to the 30% response to antidepressant medications. You have a high probability of getting them better, a little bit better within hours, and a lot better within a day. And that could mitigate, that could reduce the risks associated with depression. It could mitigate or shorten hospitalizations. It could reduce the number of work days missed, the disruption in families. This is a very exciting and potentially important idea, and it remains to be seen whether a drug like ketamine could achieve these objectives. And the other part is dealing with treatment-resistant depression, which we all know is a huge problem. And perhaps it's a way of helping people who we haven't been able to help very effectively up till now. Now, so how do you know, what is the sign that something that you've done is getting some attention, right? So when I'm a Masterpiece Theater fan, and I was watching the Inspector Lewis series on Masterpiece Theater, and they had an episode of their show, an antidepressant drug trial on the Oxford, thank you, not Yale, campus. The drug being tested is a form of ketamine. When a trial subject is found dead, Lewis and Hathaway aligned in their quiet distaste for psychiatry. Now, let's stop here for a moment. Why aren't they aligned in their deep, abiding respect and gratitude to the field of psychiatry? I don't know. Uncover a number of practical cues. The point here, and I actually have a bunch of these slides. There's a Danish TV mystery show called Dicta. There are a variety of episodes and movies. House had an episode where they had something on ketamine. Society, people out there have begun to hear about ketamine. And they are very hopeful. It's capturing an idea that psychiatry could intervene with a power, possibly. It's a little scary, as you can tell, that it's being teased in a way in this episode. But depression is really terrible. It's really, really terrible. And the idea that you could get better in a day for all those people suffering with depression is a remarkable idea. Now, there are companies already set up to exploit this opportunity. This is one called New York Ketamine Infusions. They're very good anesthesiologists. I'm a little worried about what they're promising, because I don't know how well they care for people out, for the psychiatry part. But they're already marketing a drug for suicidal. If you're suicidal, call this number. It's a really interesting thing. One of my worries is that companies are going to be set up that won't have the right kinds of protections, not the right kinds of cautions, that it will be kind of done in a sloppy way, which may put patients at risk and therefore harm the ability to advance this in a reasonable way to protect people And there's a case that I understand may involved here. So here's a case of someone named Hartman who didn't respond to treatment, went to NIMH, received 100% remission. This miraculous effects dissipated over a few weeks. Found a doctor in San Diego, maybe one of your doctors, and also New York ketamine infusions, and he got ketamine. People will find a way to get the treatment that they need. And we're beginning to see groups endorsing ketamine. So this is one of several groups. This is the Canadian Agency for Drugs and Technologies in Health. They do rigorous analyses and meta-analyses to guide Canadian medical policy, and they endorse it, but they say that it's still in its early stage. I think that's a fair assessment of where this area of work is right now. So in closing, I've presented two kinds of interesting and opposing stories. In the case of schizophrenia, I've talked about deficits in glutamate signaling and disinhibition early in the course. And that leads us to want to test drugs that enhance NMDA receptor signaling and inhibit glutamate release. I've then gone on to talk about depression, where there may be overstimulation of extrasynaptic NMDA receptors and inhibition of synaptic glutamate signaling. And as a result, you want a treatment that blocks rather than enhances NMDA receptor signaling and enhances rather than inhibits glutamate release. And this is where psychiatry is going to go. I mean, I'm not sure that anything that I've said here is true. But someday we are going to have a biology that informs the treatment, and we're going to have treatments that in specific ways address the underlying biology of the disorder. And one treatment isn't going to work for every disorder, but rather we are going to have very specific treatments, not only for specific disorders, but maybe even someday for specific patients. And that really would be a very exciting thing indeed. These are the people who contributed to the schizophrenia part of the story: Alan Anticevich, Phil Corlett, Cyril D'Souza, Naomi Dreesen, David Glahn, Greg McCarthy. I didn't really show you the computational work from John Murray and Xiaojing Wang. And the depression work, Dennis Charney, who he and I dreamed up the ketamine idea, at the VA in the evening one day. Angela Cappiello. This is not Rob Berman. That's it's actually Otto Rank. Rob looks a little bit like Otto Rank, but not exactly. Jerry Sanacora. And again, thank you very much for the opportunity to come down and share these wacky ideas with you. Thank you.
Well, thank you, John, for just a wonderful talk and so rich. I'm sure there's some questions. Would you mind
Please
addressing them? Barbara Perry.
Thank you for this very thorough, comprehensive.
Hang on, you're getting the microphone.
Okay. It reminds me of Bob Post's work many years ago on mood disorders, how differential effects of lithium versus antidepressants may be effective. I'm wondering, based on clinical phenomenology, that young males are at increased risk for schizophrenia,
right.
Women after puberty for depression, but females may develop a late onset psychosis.
Yes.
Does that inform your, the pathophysiology? And my second question is, in terms of the depression and the treatment, other than the stimulants, the other rapid onset antidepressant we have, of course, is sleep deprivation,
right.
wake therapy. But you lose the effects afterwards, and you have to supplant. And I'm wondering if that gives you any insights into its pathophysiology in relation to ketamine.
Sure. So the first question was about sex differences, and the second question is about sleep deprivation. So there are sex differences in ketamine response. So women have, in our experience, it's not a very big effect, you need a large number of people to detect it, are a little bit less sensitive to ketamine. In other words, they have a kind of built-in tolerance to ketamine, a little bit. And the story that I've been very interested in about that, which I've wondered whether it could be harnessed in some way, is that estrogen, when given intermittently, not constantly, only when intermittent exposure, like menstrual cycling, causes a sprouting of dendritic spines and increased in NMDA receptor expression. And it doesn't last. In other words, if you keep steady estrogen, those spines go away. But there may be ways to harness estrogen as a way to enhance cognition in this pulsed way in combination with cognitive remediation or other strategies that depend on synaptic integrity to produce the beneficial effects. And there might even be synergies between estrogen and the glycine pro-NMDA signaling, because again, the estrogen's only increasing the availability of NMDA receptors on dendritic spines. It's not stimulating them. So that some other strategy might be useful, like cognitive remediation or glycine to enhance that. With sleep deprivation, actually, there's another Krystal who could tell you probably more about sleep deprivation than I. But there are some studies trying to extend sleep deprivation. Biff Bunny's group at Irvine has used, I think, D-cycloserine or something like that in combination with sleep deprivation to try to extend that effect, and has had some initial success. I think it's still early days in trying to figure that out. I assume that there's a lot going on with sleep and a lot that gets disrupted, but I don't really know the answer to that.
Okay, so you are proposing there is opposing biologies between schizophrenia and major depression.
Yes.
So
it's not a. I omitted the parts that are not opposing, but that's okay.
Okay. So well,
I'd say dimensional and categorical distinctions.
Okay. So I think can you comment on the Scottish DISC1 family?
Yes.
In that family, the translocation co-aggregated with schizophrenia and major depression. So
yes.
How can the same genetic mutation generate this kind of
yes.
Almost opposite?
Yeah. So this is a great story, and one that I think about a lot. So if you don't know this family or this story, there's a family in Scotland that has a translocation in a gene that's called disrupted in schizophrenia 1 or DISC1. And in this family, there are increased numbers of people with schizophrenia. Now, I think there are 5 or 6 people with schizophrenia. 7? Okay, 7. Then there's someone with alcoholism. There's a couple people with depression or bipolar disorder, someone with borderline personality disorder, and there are some healthy people. And they all have the same genetic mutation. And so how do we understand the heterogeneity of the clinical phenotypes generated from this gene that we would think would have a big and somewhat specific effect on the risk for schizophrenia? So and why would you have? How could you have depression and schizophrenia if they have opposing biologies, which is really your main point. So the first is to acknowledge there's something we don't understand about how you go from genotype to phenotype in this family that is so clearly illustrated. And we don't really know the underlying biology of how DISC1 is expressed. But the animal models for DISC1 are very complicated. So animals that have knockouts of DISC1 don't show the typical phenotype of schizophrenia in terms of response to NMDA receptor antagonists. So humans with schizophrenia, garden variety of schizophrenia, whatever that is, if you give them an NMDA receptor antagonist, they're hyperreactive to NMDA receptor antagonist effects. Their schizophrenia symptoms worsen. They may be upset for several days after the test. Animals with DISC1 knockouts, and I qualify this by saying that not all DISC-1 knockout animals look similar. But animals with DISC-1 knockouts, to the extent that they show altered response to NMDA receptor antagonists, have reduced sensitivity to the cognitive and behavioral effects of NMDA receptor antagonists. So there's something, in other words, it suggests that the DISC-1 knockout animals either have tolerance to the effects of NMDA receptor signaling, in other words, an upregulation in something, or they have an abnormality that has occluded the expression of the response to NMDA receptor antagonists. But since the underlying biology of those DISC-1 knockout animals hasn't been fully worked out and linked to the NMDA antagonist response, we don't really know which one that is. So my guess is that because of how DISC-1's involved on inhibitory interneurons, my guess is that it's hitting one element which has been described to be similar in depression and schizophrenia, which is GABA deficits in schizophrenia, GABA deficits in depression. And as opposed to hitting the dimensional aspects of those disorders, as opposed to hitting the categorical biology of those disorders. That's my hypothesis, but I don't think the biology is far enough along. I don't know if you have anything to add to that.
Well, I just have one comment. Yes, actually, the Scottish translocation is not just the disruption of the DISC-1 gene. So initially, they think there is a translocation between chromosome 1 and 11. The DISC-1 was disrupted on chromosome 1, and they report there is no genes on chromosome 11. Actually, there is the genes. So they are actually the fusion genes between two different genes. So it's not simple. Their family is not a simple DISC-1 disruption.
Yeah. Thanks.
Thanks for the wonderful presentation. Two disparate aspects. I wanted to address the rapidity of onset with ketamine, which is, of course, what is so appealing and so remarkable for the treatment of depression. But in the long-term care of depressed patients, what are we going to do? Because do you envision ketamine as a long-term treatment, or is it a way to buy time to get some other treatment in place?
I think we don't have the definitive answer on that. So I would say that the data that we have from people who have treated patients long-term, including people here, Pierre Blier in Ottawa and some other groups where we have data now for 3 years of weekly or biweekly, or in some cases even monthly treatment, is that patients can tolerate that. They don't necessarily develop big time tolerance to that. I see David nodding on this, and it seems to be very safe. And what's interesting to me from a safety point of view is how different ketamine abuse is from intermittent ketamine. So the ketamine abusers, they use more and more frequently, more and more drug, more and more frequently. And so the ketamine abusers we've worked with in Taiwan and in China, so we give 35 milligrams about for the antidepressant dose of ketamine. And there are people who are taking 6, 7, 8, 10 grams of ketamine intranasally a day. And so ketamine abuse and ketamine treatment in terms of dose is really just completely different. So the intermittent low-dose ketamine, as far as we can tell, is reasonably safe. But I like the idea. So this is kind of like sensitizing people to ketamine and producing hopefully adaptations that last longer and longer. And there are examples, probably the most elegant, not with ketamine, but in non-human primates from Stacy Kastner's and Patricia Goldman-Rikich's work, where sensitizing regimens of drugs can, over time, have more and more sustained profile of efficacy. So it may be that we discover over time that ketamine becomes a better and better treatment the more and more you go, if you can tolerate it. That being said, there is this interesting property of ketamine of enhancing synaptic plasticity during that time. Growth of dendritic spines. Ron Duman has shown that animals extinguish fear more rapidly after ketamine exposure, et cetera. So it may be that ketamine, as you say, makes people more responsive to both behavioral and pharmacologic treatments over time, and that it becomes unnecessary, which would also be fine. So I think these are all options that haven't really been worked out. But I think at a minimum, it's safe for many people long term.
Hi, thank you again for the wonderful talk. I'm going to pick up on the question from Shinjin about the genetics and the Scottish family and ask about bipolar disorder. Because bipolar disorder may be more genetically similar to schizophrenia, and yet it looks like it's responsive to the ketamine effects in antidepressants. And so are there any lessons about bipolar disorder we can take away from ketamine?
Yeah. So it's really the group that hasn't gotten ketamine are the psychotic bipolars who share the most genetic loading with schizophrenia. And that'll be an interesting group. I don't know whether that it will work so well in that population. We'll just have to see. The people who've got it are so far have been bipolar depressed patients who share a lot of the biology with major unipolar major depression. I think bipolar disorder is a disorder that may turn out to break down into two disorders, ultimately, given what's coming out of the genetics, and so that would be ketamine might be one of those things that separates those two groups, but we just have to see.
Thanks for this really wonderful talk. It's really nice to see how this evolved over time and to see it all laid out like that. One thing we didn't talk about yesterday when I had the fortune to meet with you was the anxiety story. So my work, I think it works better for anxiety than depression. I have 2 cases of full-blown panic disorder that remitted. And if you look back, some of the papers had secondary analyses on anxiety, and it looks like there is an anxiety effect in those people, in those papers that studied it. So I wondered if you could comment on your thoughts on the anxiety issue.
Yeah, I think that the anxiety story is probably there as well, and for all the reasons that you say, which is that it hasn't received the focus that the depression symptoms have received. It is going to be increasingly a focus as ketamine is used for post-traumatic stress disorder in the studies of post-traumatic stress disorder, there's this interesting story that when people ask me about the comorbidity of anxiety and depression, I always think about Ken Kendler's studies where he suggests that genetically GAD and depression are basically the same disorder. It just depends on what obstacles life presents to you in your life course. So it wouldn't surprise me if it was useful. I think the issue is the risk benefit. So if someone has panic disorder, the chances that you can get them better with currently available medications are pretty high, or GAD in general. And that's not the case for major depression. The chances of getting people into remission with major depression much more protracted, much longer and harder, so that the risk-benefit equation is typically more favorable for ketamine in the mood disorders. But we all know that there are people who have refractory anxiety disorders where that anxiety disorder is as crippling, terrible, disabling as anything you could imagine. And I highly recommend everybody read a book called My Age of Anxiety by Scott Stossel, who is an editor of the Atlantic magazine, who both tells his incredible story of treatment refractory panic disorder, as well as reviews the history of psychopharmacology in a really well-written way that illustrates just how bad panic disorder can be and how it makes a very strong case for a favorable risk-benefit profile for anxiety disorders with drugs like ketamine. So I think that will get there, but probably it'll be reserved for more treatment refractory cases. Yeah. Yeah.
Thank you for a wonderful presentation.
Thanks.
Your work makes a compelling case that psychiatry can and will move towards precision medicine, being able to identify the right treatment for the right person at the right time. I wonder if you could comment a bit on the role that biomarkers might play in improving the prediction of which individuals are most likely to exhibit a response, or to use biomarkers to monitor a response to these kinds of new treatments.
Yes. So, I was just thinking that precision medicine seems too lofty a title to describe. So I've just described one distinction between two disorders, but clearly, we've all had patients sit down with us and say, "Is this drug going to work for me, or are you experimenting on me?" And we all have that moment where we think, "Well, how am I going to make this sound better than it could?" And because every treatment of every patient is an experiment, and keeping that in mind is really the essence of academic medicine. Not pretending we know more than we do, trying to learn from each patient and to carry that knowledge forward. And I'm not convinced. I don't find the biomarkers that have been used so far to predict ketamine response compelling. I think what the biomarkers that people have used so far have done are to identify people who fail to respond to traditional antidepressants. Higher levels of anxiety, bipolarity, inflammation, all these are markers of poor antidepressant response. But they don't appear to be markers of poor ketamine response yet. So what we have done is essentially biologically remind ourselves that ketamine is a relatively more effective antidepressant in treatment-resistant depression populations. I'm not sure we have anything that's a useful positive predictor for ketamine yet.
Yeah, hi, I work with Scott at the Cancer Center with people with advanced illness, and was really interested to hear more about two things. One, the intranasal use in terms of the nuts and bolts and how I could go do that this afternoon.
Yeah.
And then the second one was looking at what you've observed with patients who use ketamine chronically for pain in terms of, that's a multiple day dosing for long periods of time in terms of development of any issues.
Yeah. So all of us in the ketamine depression field have followed the pain literature pretty carefully because in that area, there's a lot of clinical experience, a lot of publication about long-term ketamine safety, where it really seems quite favorable and has been quite motivating for the long-term studies of ketamine as monotherapy or long-term adjunctive therapy for psychiatric conditions. I really think that the work that you guys are doing to use ketamine in context of comorbid pain and disorders, which could be very relevant, say, for PTSD as well, where we have a lot of chronic pain combined with mood disturbance, is really wonderful. And because it avoids, it's not only what ketamine provides, which is pain control and antidepressant response, but it also what it avoids, which is the need for escalating opiate use and the relatively poor pain control that some of these people get into once they become very tolerant to opiates. So I think this is really very exciting and very important work that you all are doing.
We have two more questions and we'll have to wrap it up. Yes.
Yes, thank you again for this wonderful talk. I was curious, you've mentioned the use of ketamine, OCD, PTSD, anxiety. Are you aware of any research being done into using it for the treatment of eating disorders?
Eating disorders. I'm not aware of use yet for eating disorders. We have tried to use memantine to look at it as a treatment for alcoholism. And memantine is, we've used it in relatively low doses. And we find there's a negative study by Suzette Evans at Columbia. And in our work with memantine, what we find in the laboratory is that it reduces craving for alcohol. It reduces the stimulatory effects of alcohol. But if you put people in the laboratory and give them opportunity to drink, it doesn't reduce their drinking. Interestingly, naltrexone doesn't reduce craving for alcohol in our hands, doesn't reduce the stimulatory effects of alcohol, but if you put people in the laboratory and you give them naltrexone, it reduces their drinking. So we're actually doing a study now. We interpret that, by the way, as to say that maybe memantine is targeting the reward-related aspects of consumption, and the naltrexone, the more drinking or habitual aspects of the consumption. And so we're actually testing now the combination of naltrexone and memantine. And if that combination is helpful for the alcohol people, it may be helpful for other addictions, including food addiction, which are some kinds of eating might be relevant to some kinds of eating disorders. I don't personally know enough about. I know there's a lot of work on anorexia nervosa and bulimia here. I just don't know those fields well enough to be able to comment on that.
Jon, thank you very much for such a stimulating talk. There are many, many questions we like to ask, but one thing which I like to pick up today, it struck me, you mentioned about the schizophrenia is preventable, but you have to find a critical time.
Yes.
Early.
Early.
Yeah,
Right.
how to do that? Do you have any ideas?
Well, so this gets to an issue about primary and secondary preventions.
Yeah, that's right.
And my suspicion is, and I think that I don't know if I'm being optimistic or pessimistic. My suspicion is that if we want to intervene in ways that fundamentally change the course, we probably have to intervene before there are any symptoms whatsoever. In other words, before the high risk or limited symptom or whatever you want to call that state is where people are seeking treatment. But when they're in elementary school and their only stigmata may be, like I'm thinking about the Erlenmeyer-Kimling studies, this non-specific, really, attention dysfunction, certain kinds of very subtle cognitive impairment that have a very low probability of resulting in schizophrenia. And probably are not specific for schizophrenia, but may be markers of a variety of dysfunctions. And because since we're not treating Huntington's chorea, so we don't have a gene, even in the rare variants, that increase your risk 10 to 20-fold for schizophrenia. Do we have we reached a threshold there, where we can intervene pharmacologically in asymptomatic people? I don't know if we're there. I don't know if culturally we're there. Probably not. But we're not far from a place where the idea of cognitive enrichment for children who are broadly understood to be at risk could be very helpful. And it's worth a cultural experiment, I think, in going broad and going early, very early. And there would be ways to enrich the sample for genetically enriched sample with very mild cognitive impairments of the kind that group described. I mean, that would be the kind of way I would do it. That I would probably say is secondary prevention, because you're not fixing the underlying etiology per se, but you're hoping, and this is the optimistic part of it, you're hoping that if you can target these circuits early, while they're still relatively intact, that you may be able to shape the course in a way that's ultimately more favorable. And it may be ultimately more favorable for a variety of conditions. But that's how I kind of think about it at the moment. I think in our traditional prodrome or in the period leading up to it, that you would target more intensive neuroplasticity modulating approaches. Could be GlyT, could be D-serine, could be transcranial direct current stimulation could be, there are a variety of approaches that one might think for trying to intervene at that later stage.
Well, join me again in thanking John very much for this wonderful, elegant presentation. We have a tradition in this department that we honor our grand round speakers by welcoming them to the Department of Psychiatry at UCSD. In your case, this is especially important. We want to reduce any sibling rivalry because Andy does have one of these hats. Yeah, but you could wear it backwards and he could wear it forwards, whatever
you gave it to him first.
He got here first. What can I say? But thank you so much. Wonderful.